everybody. So I'm today in Texas, in Houston, in Baylor College of Medicine, and I'm with Pawel Stankiewicz. Could you please introduce yourself?
Hi, I'm a full professor here in the Department of Molecular Human Genetics at Baylor College of Medicine, the Texas Medical Center in Houston, Texas, where I have my own research laboratory. For the last 15 years, working on genetics and genomics of pulmonary vascular disease and lethal lung developmental disorders in particular.
Thank you. So you joined TBX4Life about a year ago, and from that time I learned that you spent a lot of time researching different developmental diseases like FOXF1, for example, based, and also then later TBX4. When we talk about TBX4 in particular, could you please tell us, in your opinion, what it makes it special, what it makes it important, what research about TBX4 can bring maybe to the understanding of the lung diseases, to understanding of these anomalies which happen, and also maybe to ultimately to the treatment approaches and to the families?
Sure. So just short background, when we started this research about 15 years ago, with discovering the FOXF1 as a causative gene for alveolar capillary dysplasia, one of these rarely diagnosed lethal lung developmental disorders, we started to receive samples from all over the world, because these are rare diseases, for testing, for diagnosis, and some of these samples actually turned out not to be alveolar capillary dysplasia by re-evaluation of our experienced pathologist. And we then systematically started to investigate this specimen, these samples, and we identified TBX4 gene and mutation and deletions or defects in this gene as causative for acinar dysplasia and congenital alveolar dysplasia. These are very, very rare lethal lung developmental disorders. So we discovered this gene 5 or 7 years ago, and then described the clinical consequences in a few patients with deletion, the point mutation of this gene. Mm-hmm. And we also described the phenomenon, a very interesting genomic phenomenon, which we call represent a kind of complex compound inheritance of TBX4 in the families, where the same variant, same defect in TBX4 segregates in multiple individuals through many generations, manifesting with very different clinical phenotype, which we describe as variable expressivity or incomplete penetrance. So again, some children born and die with the lethal disease few days after birth, and some being mildly affected pulmonary hypertension, and some family members with this defect being completely unaffected. That's fascinating from a genetic standpoint.
I understand, yeah. I think that it resonates also to what we see in our community, because definitely, like I told you, we have all the scenarios possible, right? So, and one of those kind of like incomplete penetrance, I'm just example of that, right? So where my son is severely affected and I hopefully not affected, yeah. I think that is the biggest mystery for our community, and we struggle to understand kind of this knowledge about TBX4 mutation in the family, what it brings to us, right? So like, does it mean that everybody has, not everybody, what it depends on, etc. Besides that, of course, what it makes it different, really?
Well, so we try to, like I say, understand the underpinning, the molecular basis of this phenomenon, and we fortunately have now the newest technologies that allow us to screen the whole genome of the affected and unaffected. So we ideally like to study family trios with the affected child or deceased child sometimes and the parental samples, using this whole genome sequencing technologies, and then try to analyze, interpret, and finding the explanation for this variable expressivity. or incomplete penetrance that I mentioned.
Mhm.
And so we found that TBX4 represent, with another downstream gene we call the FGF10, represent a different biochemical pathway in lung development, which is kind of independent from the focus of one that I described, and we are in particular interested in their interactions, how they interact, how they affect the human lung development.
I see. So yeah, I think that anything what we can do from our side with respect kind of like so that you can continue your research or expand your research. So when you talk about trios, my understanding of that it's kind of talking about blood samples of affected children, for example, and parents where that is possible, right? So this is probably one of the things which over the time we can expand. Is there some particular message or kind of like some particular issue which you'd like to kind of share with TBX4Life and the community?
Well, first of all, I really want to thank and appreciate the effort of Anton and Eric, funding and spearheading this TBX4Life initiative, which helped to build a network of investigators representing different fields of science and medicine, including developmental biologists, geneticists, pulmonologists, cardiologists. Now, joining forces, we can really accelerate advanced science research and make a difference. So we really appreciate this networking and building the community. And because of the rarity of these diseases that we, especially the little lung developmental diseases, now we have access to more very precious samples that we can study in detail and compare. And that is really invaluable for our research. So thank you again for all your efforts.
Thank you very much, Pawel. Yeah, this is how we have this even on our first flyers. Every case matters.
Every case matters, absolutely.
Thank you for being a part of
My
our team.
pleasure. So keep it up.